Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Amplify Your Viral Vaccine Production with CRISPR-Cas9 Engineered Host Cells, presented by Dr. Elizabeth Turner-Gillis. Dr. Gillis is a scientist at ATCC. In this presentation, Dr. Gillis will provide an overview of how viral vaccines are manufactured in large-scale tissue culture systems using historical cell lines. Then, Dr. Gillis will describe how ATCC used CRISPR-Cas9 gene editing techniques to increase the viral production efficiency of some of those cells. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I would like to welcome Liz Turner-Gillis.
Hello, this is Liz Turner-Gillis. I'm a scientist with ATCC Cell Biology. I work with ATCC's CRISPR-Cas9 engineered advanced cell models, and today I'm going to be talking to you about our CRISPR-Cas9 engineered host cells for enhanced viral vaccine production. First, a short introduction to our organization. We were founded in 1925, and we are a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and services center in Gaithersburg, Maryland. We're the world's largest, most diverse biological materials and information resource for cell lines and microbes. In addition to being the gold standard for commonly distributed cell lines, we also have an innovative R&D program that uses technologies like gene editing and next-gen sequencing to develop advanced cell models. We're going to be talking about some of these gene-edited advanced cell models today. This webinar has three parts. First, I'm going to give a short overview of both viral vaccine production and CRISPR-Cas9 gene editing and how this technology can be used to enhance viral vaccine production. Then in the second part of the talk, I'm going to focus on the use of Vero cells for viral vaccine production. Then I'll talk about how we've used CRISPR-Cas9 to enhance the viral production capacity of Vero cells and how we ensure that our enhanced viral production Vero cells meet ATCC standards for quality and reproducibility. I'll also go over the functional validation of increased viral production in gene-edited cell lines that's been performed by our R&D team. Finally, in the third part of this webinar, I'll talk about the use of MDCK canine kidney cells for the production of the flu vaccine and how we've used the same strategy to enhance the ability of the cell line to produce influenza virus. So in the first part of the talk, concerning viral vaccine production and gene editing in general, I'll first give a short overview of how viral vaccines work. I'll then talk about how they're manufactured in host cell tissue culture. I'll then talk about some of the cell lines used for viral vaccine manufacturing and the opportunity we see here to increase the viral production capacity of these cells. I'll talk about CRISPR-Cas9, which is the technology we'll be using for cell line modification. I'll talk about how we'll be modifying the host cell antiviral response using CRISPR-Cas9. I'll talk about how we'll be using the non-homologous in-joining error-prone DNA repair mechanism in conjunction with CRISPR-Cas9 to perform a gene edit. And then I'll talk about the development of enhanced viral production cell lines using CRISPR-Cas9. So first, an overview of viral vaccines in general. Some of the most commonly used viral vaccines are listed over here on the right. These include the flu shot, the MMR vaccine, chickenpox vaccine, polio vaccine, HPV, vaccines against viral hepatitis like Hep A and Hep B, the rotavirus vaccine, and the rabies vaccine. Viral vaccines work by introducing your immune system to a virus or part of a virus in such a way that it enhances your immune system without actually making you sick. There are a variety of strategies for doing this. Some viral vaccines consist of whole viruses that have been inactivated or killed in some way. Other viral vaccines use whole viruses that are alive and active, but weakened in such a way that they don't make you sick. Some viral vaccines are made up of just some part of the protein that's on the outside of the virus, and those can be made either synthetically or purified from a biological protein system. Viral vaccines can also be given in a more roundabout way by introducing some of the genes for the virus of interest in the form of raw DNA, viral DNA delivered in a bacterial vector, or viral DNA delivered in the packaging of a different unrelated virus. What then happens is the viral DNA behaves as it does when it's given to your cells by the virus itself and starts instructing your cells to construct viral proteins, which are then recognized by your immune system without making you sick. Given that, most of the common viral vaccines listed on the right are either whole inactivated, live attenuated, or viral vector-based vaccines. And this is important because of the way these types of vaccines are manufactured. So a lot of vaccines are produced in chicken eggs, and then the viral particles or pieces of the virus are purified in large scale from those chicken eggs. But a lot of other vaccines are manufactured using a large-scale tissue culture system. In these systems, the virus or viral product of interest is mass-produced from a host cell line and then purified. To maximize the amount of virus produced, the host cells can be adapted to grow in suspension tissue culture, or if the cells are anchors dependent, as in the case of Vero cells or MRC5 cells, the cells are cultured on microcarrier beads, which are then cultured in suspension. An important thing to note here is that while every aspect of large-scale viral vaccine production in a host cell tissue culture system has been optimized to maximize production output, for the most part, a small handful of historical cell lines are being used as host cells. These cell lines include MRC5 and WI38, which are both human lung fibroblasts isolated in the 1960s, Vero, a non-human primate kidney epithelial cell used for the propagation of a wide variety of viruses, also since the 1960s, and MDCK, a canine kidney epithelial line isolated in 1958. While there's been a lot of work done in terms of clone selection, media conditions and timing, and culture conditions and technologies for these cell lines over the years, very little innovation has been made in terms of introducing new viral production cell lines or using genetic manipulation to increase the viral production capacity of the historical cell lines themselves. As a distributor of these historical cell lines, at ATCC, we see an opportunity to help make viral vaccine production more efficient. By modifying historical cell lines currently used for viral vaccine production, rather than introducing new cell lines, manufacturers of vaccines can increase their output and lower costs while using the same scale-up and purification systems they've already developed. There are also fewer regulatory hurdles to using a modified version of an existing cell line than to introducing a completely new cell line into the viral vaccine production workflow. The technology we're using for cell line modification in this case is CRISPR-Cas9. This technology can be used to permanently change the genetics and characteristics of a cell line. It works by locating specific gene sequences inside the nucleus of a cell and cutting the host cell DNA at that location. Once the cut is made, the nucleotide sequence at that location can be changed to something else, or nucleotides can be added or removed. These genetic changes are permanent and can change the characteristics of the cell controlled by the modified gene. Our specific strategy for increasing viral production capacity of our host cell lines involves disruption of STAT1 signaling. STAT1 is a key signaling molecule in the interferon signaling pathway. Interferon signaling is part of the cell's innate immune response. Cells respond to viral infection by secreting interferon molecules, which are then detected by cell surface receptors in neighboring cells. This signal is then carried to the nucleus by STAT1 and STAT2, which then activates a series of interferon response genes, which then themselves constitute an antiviral immune response. If the STAT1 gene is disrupted by CRISPR-Cas9, no STAT1 protein will be produced, and interferon signaling will not be carried to the nucleus, and the cells will not be able to affect an antiviral immune response. Cells defective for antiviral immune response should then be more permissive to viral infection and should produce more viruses easier. Cells have two DNA damage repair mechanisms that can be used to repair the double-stranded break made by CRISPR-Cas9. Our strategy is to design and construct CRISPR-Cas9 reagents targeting the beginning of the STAT1 gene, and then use an error-prone DNA repair mechanism called non-homologous injoining to repair that double-stranded break. Because this DNA repair mechanism is error-prone, small insertions and deletions of one or more nucleotides will frequently be made at the site where CRISPR-Cas9 cut the gene, resulting in an out-of-frame mutation. Because an out-of-frame mutation will be created in the beginning of the STAT1 gene will then be disrupted. In general, our workflow for generation of enhanced viral production cell lines is as follows. We first evaluate the host cell lines for the ability to undergo CRISPR-Cas9 gene editing. We perform transfection evaluations and cutting frequency assays. We then select genes that are likely to affect our phenotype of interest, in this case, STAT1 for increased viral production. We then perform CRISPR-Cas9 gene editing, resulting in a pool of edited and unedited cells. We then test that cell pool for the efficiency of CRISPR-Cas9 gene editing and a preliminary biofunctional testing for increased virus production. We then perform single-cell cloning, grow up our clones, and screen those clones using Sanger sequencing and biofunctional screening for increased viral production. We then select our top clones and perform additional sequence verification, transcript sequencing. We look at the protein expression profile to determine if STAT1 protein expression is present. We perform some off-target CRISPR-Cas9 gene editing evaluations that I'll be talking about later. We do stability testing to ensure that the genotype and phenotype of interest is stable over time. We look at the morphology and cell growth properties of the resulting gene-edited clones to make sure that they're similar to that of the parental cell. And we perform additional biofunctional evaluation, resulting in enhanced viral production cell models that are then available for sale. Now we're going to specifically talk about what we've done with Vero cells. So as I mentioned, Vero is an adherent epithelial cell line established from the kidney of an adult African green monkey in 1962. Vero cells are unusually permissive to infection by a wide variety of animal virus because they're partially defective for interferon production. For this reason, they're one of the most commonly used cell lines for cell-based viral vaccine production, and they've received worldwide regulatory acceptance. There's an abundance of studies on the production of viruses in Vero cells, and this data includes things like influenza, vaccinia, and polio, for which vaccines are currently being produced. Our specific CRISPR-Cas9 gene editing workflow looks like this. We first design and construct plasmid constructs for expression of Cas9 and puromycin resistance, as well as guide RNA targeting the beginning of the STAT1 gene for African green monkey Vero cells. We then transiently transfect these plasmid constructs into the wild-type Vero cell population. After the transfection, we perform a short puromycin selection to enrich the pools for cells that have been transiently transfected with CRISPR-Cas9 construct. Then after a short recovery period from puromycin selection, we do single-cell sorting and clonal expansion. We then assay our resulting CRISPR-Cas9 edited Vero clones for STAT1 gene disruption. And again, because these Cas9 and puromycin constructs are transiently transfected into the Vero cells, they will not be included in the genome of the resulting clone. So the STAT1 knockout modification that was made in Vero cells is a 199 nucleotide deletion in both chromosomal copies of the STAT1 gene. This is a Sanger sequence alignment showing the reference sequence for the wild-type STAT1 gene in African green monkey Vero cells, our Sanger sequence of unmodified Vero cells, followed by low and high passage STAT1 edited cells. You can see that 199 nucleotides have been removed, resulting in disrupted STAT1 gene sequence. Disruption of the STAT1 gene in this way results in a functional STAT1 protein knockout because the DNA from the STAT1 gene has a large section missing from it, which results in a shorter truncated messenger RNA, which results in a STAT1 protein that, while the beginning of it looks like normal STAT1 protein, after the deletion, all of the amino acids are out of frame, and so they don't match the normal STAT1 protein. In addition to being out of frame, it's also truncated because there's the presence of a premature stop codon. This truncated, aberrant, non-functional protein is then recognized by the cell as aberrant and rapidly degraded, resulting in a loss of functional STAT1 protein. This is an immunoblot using an antibody targeting the N-terminuses of the STAT1 protein, showing that we have loss of STAT1 protein expression in our STAT1 knockout clones. On the left, you can see the STAT1 protein is detected nicely in the parental wild-type Vero cells, while in the STAT1 knockout proteins, or STAT1 knockout clones, no functional full-length protein is detected by immunoblot. We then use PCR genotyping to analyze the STAT1 gene. In this case, we've extracted total genomic DNA from wild-type and STAT1 knockout Vero cells, and we've extracted total RNA from STAT1 knockout and wild-type Vero cells and reverse transcribed to cDNA, then amplified across the region targeted by CRISPR-Cas9. In the wild-type Vero cells, we detect an amplicon of about 1,200 base pairs in the genomic DNA, while in the STAT1 knockout cells, we detect an amplicon of about 1,000 base pairs, detecting the 199 nucleotide deletion in the STAT1 gene. We see the same thing in the cDNA. A much shorter product is detected in the targeted region. The method we're using to measure viral production is called TCID50. This is an endpoint dilution assay that measures the 50% tissue culture infective dose. This is a two-step viral infection assay. In the first step, we take wild-type Vero and STAT1 knockout Vero cells, and we infect with the same concentration of a virus of interest. We then allow that infection to run its course, collect the viral supernatants produced by those two cell lines, and then reinfect them onto an evenly seeded 96-well plate containing wild-type Vero cells. We infect the 96-well plate in a 10-fold serial dilution in triplicate, then allow the second infection to run its course, and then score the wells as positive or negative for viral infection. This scoring is then used to calculate the concentration of viral particles found in the initial viral supernatants produced by wild-type and STAT1 knockout Vero cells. For our preliminary biofunctional screening, we use a GFP Sendai virus to score the wells positive or negative for viral infection easily. This is an example of 18 wells of the TCID50 serial dilution plate that we use to calculate the viruses produced, with experimental replicates running horizontally across the top and technical replicates running up and down. And you can clearly see that on the right, the supernatants from the STAT1 knockout Vero cells have more viral infection than those from the wild-type Vero cells. Once those plates are scored as positive or negative for viral infection, we then calculate the amount of virus present in the initial viral supernatants, and we find that there's about 10 times more infectious GFP Sendai virus coming from the STAT1 knockout Vero cells compared to the wild-type Vero cells at 72 hours post-infection. Now, because nobody really is interested in vaccinating against GFP Sendai virus, we then test the biofunctional phenotypes of our STAT1 knockout Vero cells using a model clinical strain. In this case, we're using dengue virus. Dengue virus is traditionally thought of as a tropical disease. It's spread by the Aedes aegypti mosquito. It's not the same mosquito that spreads the malaria virus, but it is the same one that spreads Zika, chikungunya, and yellow fever. Now, unfortunately, in the last years, the range of this mosquito has been increasing north and south, and so we predict that demand for vaccines against these types of diseases will increase in the future. And because the dengue virus isn't labeled with a GFP tag, we have to use an antibody to detect the virus. So this is an example of viral infection of STAT1 knockout and wild-type Veros with the same concentration of dengue virus, which has then been allowed to run its course for 7 days and then stained with an anti-dengue antibody to detect the virus. And we can already see that even in the initial infection, there's about 3 times as much GFP signal indicating the presence of dengue virus in the STAT1 knockout cells compared to the wild-type Vero cells. We then take the dengue viral supernatants produced by these 2 cell lines and perform a TCID50 assay to measure the concentration of virus produced. This is an example of a TCID50 96-well plate stained with anti-dengue antibody. And in this assay, we see, again, about a 10-fold increase in the amount of dengue virus produced in the STAT1 knockout Vero cell compared to the wild-type Vero cell. Another way we measure the amount of dengue virus produced in the STAT1 knockout cells is by looking at the number of viral genomes present inside the cell at the end of the viral infection. Normally, when a cell mounts an antiviral response due to interferon signaling, the viral genomes will be rapidly degraded. But in this case, the cells can't respond, and so there should be more viral genomes present inside the cell. We use real-time PCR to detect the number of viral genomes and compare that to the number of housekeeping genes, which represents the amount of host cell genomes. This should give us an approximation of the amount of viral genomes per cell. In this assay, we see about a 30-fold increase in the amount of dengue genomes per Vero genome in the STAT1 knockout cells compared to the wild-type Vero cells. Another way we look at the biofunction of our STAT1 knockout cells is by doing an interferon signaling experiment. In wild-type Vero cells, if we pre-treat the cells with exogenous recombinant interferon gamma to warn them of a viral infection and then infect both treated and untreated cells with GFP Sendai virus, we should see a reduced amount of GFP Sendai virus infection in the cells that have received interferon pretreatment compared to the cells that have not. On the other hand, in the STAT1 knockout Vero cells, because they lack STAT1, the interferon signal won't be able to reach the nucleus and activate an interferon antiviral response. So no matter whether or not we treat them with interferon, they should give a typical amount of GFP Sendai virus infection that matches what we see in the wild-type cells with no interferon pretreatment. And this is in fact what we see in the wild-type Vero cells. We've pretreated with the interferon and looked at the amount of GFP Sendai virus infection at 48, 72, and 96 hours. And we see a reduced spread of the virus at all 3 time points compared to the condition where no interferon was given. Whereas in the STAT1 knockout Vero cells, it doesn't matter if we treated them with interferon or not, we get the same result, a typical amount of GFP Sendai virus spread. Now, after we perform gene editing to modify the STAT1 gene, we also check to make sure that no off-target gene editing has occurred. Off-target gene editing can occur when the Cas9 protein recognizes a spot in the genome that doesn't match but resembles the targeted DNA sequence. So on the top is the targeted DNA sequence in the beginning of the STAT1 gene, and listed below that are the top 12 places in the genome that closely resemble that nucleotide sequence. We perform Sanger sequencing in modified and unmodified Vero cells to ensure that no off-target gene editing has taken place at those off-target sites. This is an example of a Sanger sequencing result for a CRISPR-Cas9 off-target site. On the top, we have the reference sequence for Vero cells, and then we have all of our clones listed below that. In a red box is the potential off-target site where CRISPR-Cas9 would cut if an off-target edit was made. And we want to make sure that everything is the same as it was before. If we get off-target editing, we simply pick another clone. Next, we do PCR to ensure that the plasmid that we transiently transfected in to perform the CRISPR-Cas9 edit has not been integrated into the host cell. Because these plasmids are transiently transfected, they won't be present in the final clone. We do PCR against the CMV promoter that drives Cas9 and also against the Cas9 gene itself. Using the plasmid itself as a positive control and unmodified Vero cells as a negative control, we check for plasmid integration in all of our clones, and if we detect plasmid integration of CMV Cas9, we choose another clone. Next, we look at the morphology and growth kinetics of STAT1 knockout Vero cells and compare them to the wild-type Vero population. You can see in terms of morphology, the STAT1 knockout Veros look similar to the wild-type Vero cells, and the growth rate we measure at 28-hour doubling time versus 23 hours for the unmodified Vero cells, so slightly slower. In summary, for our enhanced viral production Vero cells, we've performed a successful STAT1 gene knockout using CRISPR-Cas9. We measure a 10-fold higher yield of both reporter GFP virus and model clinical virus, dengue2, and STAT1 knockout Vero cells relative to the unmodified cells. We measure a 30-fold increase in dengue viral genome replication in STAT1 knockout Vero cells compared to wild type. STAT1 knockout Vero cells show no antiviral response to exogenous interferon stimulation, indicating that the interferon signaling pathway has been disrupted. STAT1 knockout genotype is stable over time in the selected Vero clone, and no Cas9 plasmid integration has been detected. We detect no off-target mutations due to Cas9, and STAT1 knockout Vero cells have a similar morphology and a slightly slower doubling time than unmodified Vero cells. Now we're going to be talking a little bit about MDCK cells and how these can be used for influenza vaccine production. So over 20% of the human population is infected with the influenza virus every year. Traditionally, flu vaccines are produced in chicken eggs. This involves infection of eggs with a mixture of flu strains and then subsequent harvest of the embryonated chicken eggs to purify the viral particles. This method has several disadvantages, including time restraints and high production costs, a relatively high risk of microbial contamination because the sterile barrier of the egg is punctured for introduction of the virus, and the fact that some people can have an allergic reaction to vaccines produced in chicken eggs if they have an existing egg allergy. A mammalian culture-based influenza vaccine is a viable alternative for egg-based vaccinations, and this satisfies a need for rapid, large-scale production of flu vaccines in response to emerging flu strains. So again, we've used the same strategy here. We've used CRISPR-Cas9 to disrupt the STAT1 gene in this dog cell line. This is an immunoblot showing that no functional STAT1 protein is present at both low and high passage in MDCK cells. And again, lack of functional STAT1 protein results in lack of an antiviral interferon signaling response. This is an example of GFP Sendai virus infection at 48 hours in STAT1 knockout MDCK cells compared to wild-type cells. You can see that the virus is spreading faster in STAT1 knockout cells than it is in the wild-type cells. If we then take the viral supernatants from these two samples and then perform a TCID50 assay to measure the concentration of virus produced, we see that about 50 times more virus is produced in the STAT1 knockout MDCK cells than in the wild-type MDCK cells. Now we test using a model clinical strain. In this case, we're using an H1N1 strain of influenza A isolated by ATCC in Virginia in 2009. In our TCID50 assay using influenza A, we're staining our titer plate with an antibody that detects influenza A and scoring the wells as positive or negative for viral infection. And again, we see an over 30-fold increase in the production of influenza A, H1N1 2009, in the STAT1 knockout cells compared to the wild-type cells. To determine if the interferon response pathway is active, we treat wild-type and STAT1 knockout MDCK cells with exogenous recombinant canine interferon gamma, and then infect with GFP Sendai virus in the wild-type cells. If we treat them with interferon gamma, we see very little spread of the virus from cell to cell, whereas in the absence of interferon treatment, we see a normal amount of viral infection. On the other hand, in the STAT1 knockout cells, whether or not we treat them with interferon, we still get a normal amount of GFP Sendai virus infection, indicating that no interferon signaling due to interferon gamma is occurring in these cell lines. Now, to check if we have CRISPR-Cas9 plasmid integration in our clones, we perform PCR against CMV promoter and Cas9 gene, and we can pick this up from the plasmid and not from the wild-type unmodified MDCK cells. And again, we have no plasmid integration in our STAT1 knockout MDCK clones, indicating that there's no integration of the plasmid into the host cell genome. Next, we do our screening for off-target mutations. In the case of MDCK, there were fewer spots in the genome where CRISPR-Cas9 could inadvertently cut, so we had to screen less off-target locations. We do this by Sanger sequencing to check that no change in the sequence has occurred. Once we have a clone where we detect no CRISPR-Cas9 plasmid integration or off-target cutting, we look at the morphology of our clones and choose one that has similar morphology to the parental pool of MDCK cells and that also has a similar doubling time. In this case, the doubling time of wild-type MDCK cells was measured at 20 hours, while the doubling time of the STAT1 knockout clone was measured at 22 hours. So again, slightly slower. In summary, for our enhanced viral production host cells, CRISPR-Cas9 has been used to make precise genetic modifications. We are broadly utilizing CRISPR across our portfolio of cell lines to develop enhanced products to meet critical customer needs. Vero and MDCK cells have been modified by CRISPR-Cas9 to have a permanent increase in production capacity for a variety of viruses. Modified Vero and MDCK cells have been thoroughly characterized for viral production phenotype, stability of the STAT1 knockout genotype, lack of Cas9 plasmid integration, and the absence of off-target mutations. Enhanced viral production cells have broad applicability for basic research, production of high-titer viral stocks, and manufacturing of viruses and viral vaccines. For more information, you can visit atcc.org/vaccinedev. Thank you, and I'll now take your questions.
Well, thank you, Liz. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website, www.atcc.org. Alrighty, and for our first question, what types of viruses can be produced in STAT1 knockout Vero cells?
So for the STAT1 knockout Vero cells, you can use any virus that you're currently growing in Vero cells. So anything that you currently have in regular Vero cells or your specific type of Vero cell or Vero cell clone, you can go ahead and transition that to the STAT1 knockout Vero cells, and that'll work perfectly fine.
All right. Our next question is around growth conditions. So can the engineered viral production cells be grown in serum-free media?
So right now, the versions of the STAT1 knockout Vero and MDCK cells that we have for sale are not adapted for serum-free growth, but we're planning on adapting them for serum-free growth and releasing that as an additional product in the future. And of course, you can always do your own serum-free adaption. There's already a lot of methods available for doing that in both cell lines.
Okay, I've got another question about growth conditions. Do STAT1 knockout MDCK and Vero cells need to be maintained in antibiotic selection?
No, there's no antibiotic selection needed for the maintenance of the STAT1 knockout genotype in these cells. We did do a short puro selection during the development process, but we did the CRISPR-Cas9 gene editing by transient gene expression. And so the STAT1 knockout is permanent and there's no Cas9 construct present in the cells at this point. So there's no need to maintain it in any type of antibiotic selection.
Okay. Were there any negative side effects of STAT1 knockout in the gene edited cells?
So as I talked about previously, we did extensive off-target screening for Cas9 cutting at sites that resemble the STAT1 target site. So we didn't release any clones that had off-target gene cutting. So that rules out any negative side effects from accidental gene editing by CRISPR-Cas9. And the growth rate of both cell lines is slightly slower than the parental line, but that's much more likely to be a clone effect than any type of effect of the STAT1 knockout itself. So no, I wouldn't say we have any negative side effects of the gene editing process.
Okay. Now, are the gene edited enhanced viral production cell lines considered transgenic cell lines? I guess this is a question of terminology.
Sure. So there's additional regulatory barriers to working with transgenic cell lines and organisms. So because we didn't introduce any new genes or genes from other organisms into these cell lines, there's no non-dog genes in the MDCK cells and no non-monkey genes in the Vero cells, so they're not considered transgenic. They are considered gene edited because we've made base pair changes in the STAT1 gene, but we haven't introduced any new gene sequences, so they aren't considered transgenic cell lines.
Nice, nice. Okay, here's a question for people interested in manufacturing. Are the enhanced viral production cells available in cGMP produced lots?
Not yet, but they will be available soon.
Okay, good answer. Here's another question. How was the single cell sorting performed?
So we have a cell sorter that will put one cell in each well of a 96 or a 384 well plate. So we just had it do that and exclude cells that were in clusters or in clumps. And then we monitor the cells as they're growing up for the presence of one clone per well, and then the growth kinetics of those clones and the morphology of the clones. And then we, at that point, screen them for the knockout genotype.
Okay, here's a couple questions from the same asker. I'll start with why HEC293T host cell used for generating lentivirus instead of the Vero cells. So would the Vero cells work better? And would STAT1 knockout Vero work even better than the HEC293s?
Sure. So the HEC293Ts specifically are used for lentivirus production because they're transformed with some viral sequence that's already present in the genome of the cells. So those cells, because they already produce some viral genes, they can more easily produce functional lentiviral particles. Also for lentiviral production, usually what you're doing is you're transfecting a plasmid in for some sort of gene packaging for a gene delivery system. And HEC293Ts are one of the most easily transfected cell lines you can use. So the transfection efficiency plays much more of a role in the output than with other types of viruses where you're infecting instead of transfecting. So that's why HEC293Ts are used. I would guess that HEC293Ts would still be better for lentiviral production. Stay tuned for future HEC293T gene edited products.
Okay, here's a question around gene expression. Did you check the expression of C-gas and sting in the Vero cells?
No, we did not specifically look at the gene expression of STAT1 and interferon response elements. We were much more interested in the final resulting phenotype of viral production and as judged by the assay where we stimulated with interferon and showed no response in viral propagation rates in the STAT1 knockout cells, I would really guess that expression of those would be unaffected by interferon stimulation if we were to check.
Okay, this next question I could probably answer. Are STAT1 knockout Vero and MDCK cells available for purchase? The answer is absolutely yes. And you can go to www.atcc.org/vaccine-dev to find them through that short, friendly URL. Okay, here's another question. Did you look at the truncated protein? And can you use an antibody raised against the N-terminus to see the expression of it? I assume is what this asker is looking for.
Sure. So the antibody that we used to detect STAT1 is an antibody against the N-terminus of the protein, and we did look for that, but we don't see it. Our blots aren't really sensitive enough for the rate that it's degraded. So what I'm guessing is happening is that there's a very short N-terminal truncated protein being produced, but we can't pick it up on a blot because it's degraded so rapidly by the cell. So if it's there, we can't see it.
What about RNA levels? Is sort of the follow-up question from that same asker.
Sure. So that's actually interesting. We did check for that, and we found that actually in the STAT1 knockout Vero and MDCK cells, the level of sort of knockout aberrant mutated STAT1 messenger RNA is actually greatly increased. I'm guessing this is because the cell detects that it doesn't have any functional STAT1 protein, and so the feedback loop tells it to make more and more STAT1 protein. So it generates more and more message for that. It's just no functional protein ends up being produced as a result of that.
Interesting. Okay, now this is a question that you may or may not be able to answer. Does ATCC work on any bacteriophage therapy for treatment of infection instead of antibiotics? And how does manufacturing process of these bacteriophages differ from vaccine production?
Sure. So probably, but that's honestly the micro department, and I don't know the answer to that.
Perfectly good answer is I don't know, and honestly, I don't know either. Okay, so we'll move on to the next question. There's only about a log difference in cell growth in a commercial setting. Will it translate into a high yielding virus production? Or do you need to have 2 log difference in viral growth to be commercially viable?
Sure. So I'm not entirely sure about the manufacturing process, but from our sort of customer surveys, we think that as little as a 50% increase in production would be worthwhile in terms of switching to get increased production. Because not only do you get more virus, you can get the same amount of virus faster, or the same amount of virus with a smaller overall volume. And that could reduce cost in manufacturing either way.
Okay. Another question is, when will the webinar be online? I'm shooting to have it online tomorrow, but it may be Monday before we can prepare the file. And there is one further question about clinical trials. We do not have anything that we can report about clinical trials yet. All right. Well, at this time, we will conclude our Q&A session. I'd like to thank Liz for the excellent presentation, and thank you everyone for attending this webinar. One final word, as in previous public health emergencies, ATCC stands ready to partner with the dedicated scientists working to contain the novel coronavirus SARS-CoV-2. We're working to provide synthetic molecular standards for the virus, as well as coronavirus strains. We also have SARS-CoV-2 genomic RNA coming soon, a variety of cell lines for enhanced virus production, and custom solutions. So with that, I'd like to thank you everyone and have a great day.